All right. So today's lecture is on single-cell genomics. So here we are in the final sort of futuristic module of the course. We're talking about current research directions, and a lot of this field was not around 5 years ago. Yes. No, it will not be. Yeah. So I hope you didn't show up for the quiz. Because clearly people should not be taking this class just to get the quiz. There are other courses to fulfill your requirements that are way easier. All right, so the 4 lectures in this final module will be single-cell genomics today. Basically, how do we measure and analyze biology at the single-cell level? Then actually we're going to do, in some order, phenotype-wide association studies, basically looking at electronic health records and how do we deal with many phenotypes in the context of genetics, going beyond the sort of single phenotype approach. Then looking at cancer genomics, basically looking at sort of how all these technologies that we've learned about apply in the context of cancer. And then lastly, going from reading to writing, basically highly multiplex ways of synthesizing genomes, but also the latest and greatest technologies for editing genomes, including prime editing that was all over the news a few days ago. This is the beauty of this class. Every lecture can incorporate stuff that sometimes hasn't even appeared. All right, so the goal for today is first introduce single-cell profiling technologies, basically traditional technology for single-cell analysis, and then sort of the revolution with single-cell RNA-seq, then ways of dealing with noise experimentally, ways of dealing with noise by multiplexing, and then technologies for profiling the epigenome at the single-cell level, and then multi-omics technologies that sort of profile multiple types of data for the same cell. The second part is going to be all about analysis of these data sets. Basically, how do we cluster similar cells? How do we cluster similar genes? How do we reduce the dimensionality? How do we distinguish different cell types? How do we paint trajectories through developmental space or disease space or state? How do we complete data sets and impute missing data? How do we analyze data sets at multiple resolutions? And sort of a comparison of the multiple methods. So this is just methodology, focusing on sort of the tools. And then focusing on the applications. So we're going to look at brain, just because it's extremely important organ, it turns out. You're all using it right now. And it's also extremely diverse in terms of the cell types and the roles that they play. So it's extremely important studied at single-cell resolution. So we're going to look at initial maps of brain diversity and then brain variation in Alzheimer's and schizophrenia and bipolar, looking at somatic mosaicism in the brain, and then deconvolution of bulk data, and then deconvolution of the detail effect. Who's interested? Excited? Yeah. Awesome. All right. So single-cell analysis was not born in the last few years. Single-cell analysis has, been around ever since the microscopes were invented. So basically, being able to observe the differences between individual cells is single-cell analysis. Being able to sort of profile the fluorescence of a protein in a field of view that encompasses multiple cells is single-cell analysis. So this is not something new, but it is something that has been completely revolutionized over the last few years. So the ability to look not just at 10 cell types in the human blood, but the continuum of cell states during the differentiation of blood is revolutionary. It's something that changes our fundamental understanding of, just the whole process of blood formation, maturation, development, specification, so on and so forth. Being able to understand differences, not just between cell types, but also within a cell type. What is the diversity of responses of individual cells of the same type? Namely, every single one of these circles is not just a bunch of points in the same dot. There's a continuum in every single one of these cell types. Yes? So with better microscopes, the concept of a cell type has faded away. And you can think of single-cell RNA profiling as, the most high-resolution microscope yet. And basically, before, I think it was kind of clear what are the different cell types. We had a few cell surface markers, and you would sort cells, and you'd be like, oh, great, these are the cell types. Now, as we're starting to look at more and more resolution, these boundaries are sometimes fluid. And the concept of actually cell state versus cell type are kind of blending. Because of that continuum, we can talk perhaps at the boundaries of cell types, and then within them continuums of cell states. And it's very helpful epistemologically, like for the scientists studying them, to basically have a concept of cell type, understanding that it's fluid. Just like it's helpful to have a concept of a species, understanding that there are populations and there's, boundaries that are sometimes fuzzy. Also, the fact that you can actually switch cell type, basically that, you can reprogram cells, is something that sort of also furthers the distinction, because you could think of cell states as something that cells can, sort of fluidly go in and out of, and cell types, things that are sort of terminally differentiated. But basically, because of the ability of cells to actually switch not just state, but sometimes type, it's a more fuzzy concept. Any other questions? So again, being able to look at signaling at the single cell level, being able to look at sort of individual protein levels, and deal with very low input data sets. So what's the idea here? The idea is that if you have a bunch of cells in the brain, half of whom express a particular gene super highly and half of whom express the same gene super lowly, and you look at a bulk sample of brain, it'll be like, oh, here's the average expression. When in fact, there's simply no cell that has that average expression. So at the tissue level, you might be getting values that are simply not representative of what actually goes on in the individual cells. Everybody with me on that concept? Great. The other aspect is when you look at averages, you might say, oh, this gene is just expressed at the level of noise. But in fact, there are some cells that express that gene super, super highly. And you would just never know, because you assume that this is an average that's representative, when in fact, extreme outliers will be simply averaged out over many, many, many cells. So the traditional technologies have basically been, looking at cells, either living cells or dead cells, and then being able to detect DNA or RNA in those cells using particular probes. And some of those technologies can be real-time, that allow you to sort of look at living cells. Others are not real-time because you have to look at dead cells. And then they vary greatly in the number of things that you can observe at the same time. With these imaging technologies that I mentioned, you can observe maybe, 5 genes as, the cells are dividing, and sort of see how they're transcribed, how they're expressed, and so on and so forth. With single-cell RNA sequencing, you can now observe, thousands of cells, of genes per cell. And also, there's a big difference as to whether you need amplification or not. And then some advantages of different methods are whether they have spatial information, whether they're difficult, whether they're genome-wide, unbiased, or whether they have to look at particular probes, and they have particular constraints for each of them. So there's a huge space of these types of methodologies, and I encourage you to sort of look at the history of how we got here by sort of searching for more information for each of those cells. These are just some examples of technologies. Basically, they usually involve tagging of DNA or of RNA, using particular barcodes that you can then use to track different cells or different genes within the cell. You can basically synthesize tags that are coupled with fluorescent probes or coupled with beads, and you can sort of attach the same molecular barcode to all of the molecules that are coming from the same cell. And that's sort of the key technology with which you can then profile things at the single cell level. Namely, you might not be able to distinguish the RNAs from individual cells, but if you can tag all of the RNAs with the same barcode, when you sequence, you can computationally resolve them. Everybody with me here? Yeah. So you just basically, yes, it's a unique nucleotide sequence. And then in order to guarantee uniqueness, you just play the numbers game, because you can exponentially increase the space. You can basically generate 100,000 random barcodes, and if you profile 2,000 cells, then you're pretty confident that they're going to be unique. Any other questions? So you can basically profile mRNA in fixed cells by basically sort of building probes for the sequence of that mRNA and basically see where it is. You can multiplex it by basically adding probes of multiple colors. Basically, each of those emits a signal, a particular wavelength, and then you can sort of image at that wavelength to basically look at which of the different quote-unquote colors is inserted. And you can also use combinations of colors that basically give you a combinatorial increase in the number of distinct elements that you can detect. Everybody with me? And then, of course, the problem is that you're running out of colors. Basically, there's only so many fluorophores that you can use, and then depending on the branch length, the wavelength that they emit at, you know, there might be a lot of difficulty in being able to differentiate them. And yes, you could add more peaks, but then the signal to noise will be reduced because you will have a lot of sort of leaking across these wavelengths. So one way to do that is to basically use co-localization of multiple colors, and then the combination of all of these colors gives you additional space that you could use. And there's been a lot of development of these technologies that sort of allow you to carry out more probes, more cells, and so on and so forth. So this is the history of sort of how we got here, sort of the initial recognition that cells vary dramatically from one to the other. And that's sort of the stage at which single-cell RNA sequencing, which is really the workhorse of single-cell profiling, is coming about. So then the idea is, if you visualize using these traditional technologies in individual cells, you will see that for this particular gene, it's extremely high in some cells and very low in other cells. For that gene, you know, many cells are completely absent, and you can see this over and over again. So the question is, how do we do this systematically? And then the key technology behind this is PCR, so polymerase chain reaction. And, you know, this amplification allows you to basically profile a large number of RNA molecules simultaneously. So the idea is that you lyse single cells, and then you add some kind of primer pool, and then you basically, you know, profile each cell individually. And that's what single-cell RNA-seq allows you to do. You're basically able to dissociate from your bulk sample. You can dissociate single cells. You can capture them using either microfluidics or using basically little bubbles, little droplets that allow you to sort of capture each cell inside a droplet. You basically make droplets that are roughly the size of a cell. And then once these droplets are inserted, you can then put the same barcode in all of the RNA molecules inside that droplet. And then, you know, after you've added these barcodes, you can just sequence as if it was a bulk sample. And every single sequencing event basically tells you the barcode and therefore the cell that this came from. In addition to doing the single cell dissociation, you can also do bulk sequencing at the RNA level, at the tissue level of RNA, and then being able to compare this with that. Okay, raise your hands if you're with me so far. Awesome. So basically, after you isolate single cells, you could then use a pipette to capture individual cells. You could just plate them out and then capture individual cells. Or you could use a cell sorter. Or you could use microfluidics. Or you can use these drops. And what you end up with is a profile that basically tells you for every single cell in the columns here, what is the expression of, you know, many genes, hundreds, sometimes thousands of genes. Okay, and you can see here that some cells are extremely high for those genes and completely absent from those genes, and so on and so forth. Okay, and that basically tells you that, you know, this traditional bulk RNA profiling is completely misleading in many contexts. These technologies have been evolving dramatically. If you look at the number of single cells that you can profile, you can see this exponential scale in the increase, and sort of how the technologies have been evolving over the last few years. So basically, from the initial technology that were profiling one cell or less than 100 cells, we are now at hundreds of thousands of cells that are routinely profiled using many of those technologies. So SNARE-seq, for example, is here. This is a microfluidic separation of every single one of these cells in a different well. And then you can do all kinds of experiments in those wells. And then that technology also evolved. And then the 10x platform is probably the most widely used right now, that basically captures things into those little droplets. You basically have an oil chamber that encapsulates each of these individual cells, adding the barcodes, enabling you to do the profiling, as opposed to SNARE-seq, where you're separating each one of those cells into a single one. Raise your hands if you're with me. Awesome. All right, so this is basically the basic foundational technology. Let's now look at what are all the steps, both on the experimental side and on the computational side. So on the experimental side, the first is harvesting the cells, basically, enriching perhaps for the cell type of interest that you care about, and then doing the single cell, or harvesting cells in media, or harvesting cells from post-mortem samples and so forth. Then you prepare the cells by labeling the cells, by amplifying the transcriptome, and you can use multi-well plates and next generation technology for sequencing at the end. And then once you have all of this, you basically construct a library from either each well or from the combination of all droplets, and then you validate the quality control, and then you basically mass compile. So what DropSeq does, and that's sort of these droplet approaches, is that you isolate the cells, you suspend the cells inside droplets with barcoded beams, and then every single droplet gets one bead with a lot of barcodes. So you end up with the same barcode for all of the RNA molecules inside the same well, and then you basically sequence the rest, and the barcodes tell you what cell they came from. So how do you do this? You basically can create these beads that are attached to some PCR handle in red, and then a cell barcode, and then some UMI. What is a UMI? It is a unique molecular identifier that basically allows you to understand if something is a product of simply RNA amplification of the same RNA molecule, or whether these are multiple distinct RNA molecules. And this is extremely important because as you start amplifying, you might have amplification artifacts that basically make you think that there are 1,000 copies of this gene, when in fact, these are 1,000 sequencing reactions of the same exact single RNA molecule. So a UMI allows you to basically distinguish that. And by having, just even 4 or 5 nucleotides, you're able to say, oh, sorry, these are all the same molecules, so I can just ignore them. Everybody with me? So you can then sort of create these droplets using these combination of sort of pushing your cells through one way and then pushing these oil droplets in another way. And then what you end up with is effectively droplets that contain approximately one cell. You're basically designing the parameters of your biochemistry so that there's approximately one cell per bubble. Everybody with me? Yeah. So you put them straight at the bead level. So basically when you create your beads, you basically, when you synthesize those, you can basically say, I want GGAC and N. And the N basically says, oh, just randomly add them. Basically, as you're sort of synthesizing that pool will have, a diversity within it. Does that make sense? Any other questions? All right, so basically the SNARE-seq technology is based on this template switching idea, which is very powerful. And then the droplet technology is based on these, unique bubbles. And then the third type of technology is combinatorial indexing. And this is a very, very cool idea. So the idea of combinatorial indexing is that you never profile anything at the single cell level. It's a little crazy. You basically keep these cells intact and you push them through different wells. You basically put all of your cells in one well, which is red barcode. And then you shake them up, and sorry, you first spread them across, say, 100 different wells, each with a different color, okay? And then you repool them, and you add a different one of 100 different barcodes for the first step. And then you repool them together. Everyone has a different one of 100 different barcodes. Every single cell has a different one of 100 different barcodes in the first well. You take them all out, you shake them up again, and you now put them in a different set of 100 wells. And then each of these 100 wells adds a different barcode. And then you take them back out, you shake them up again, and you put them back in the same, or sorry, in a different set of 100 different wells, okay? Why is that exciting? Because the combination of, oh, I landed in the blue well in the first round, and in the orange well in the second round, and in the cyan well in the third round, is quite unique. And if I have 100 and 100 and 100, that basically gives me a million different tags. So it's a way of, in the case where I can suspend my cells and reshuffle them each time, it gives me a way to effectively label individual cells without ever, sort of working at the single cell level. Is everybody with me here? Okay. Raise your hands if you're following this. Okay, perfect. So you will see a series of technologies starting with the SCI, which actually is pronounced SCI. So scATAC-Seq is basically single cell combinatorial indexing. And combinatorial indexing basically means that I index in 100 wells the first round, I index in 100 wells the second round, I index in 100 wells the third round, and at the end, I have a combination of 1, 2, 3 barcodes that basically together define individual cells. Sounds good? So as we start looking at these diversity of technologies, what you would like to know is what is the throughput, how many individual single cells can I profile, what is the information content, namely how many different transcripts can I profile, and sort of how do I integrate across time, across different biological processes, whether I can perturb the cell, and what is the biological content. Are they applicable in vivo, in a particular microenvironment, and so forth. So one of the major challenges is RNA contamination. Basically, as you know, the RNA machinery that has to translate every rRNA machinery that has to translate every transcript into a protein has to have many, many copies. And as you're doing single cell profiling, the ribosomal RNA reads can sort of be quite overwhelming. And there's many ways to do this. One way is to specifically cut out our ribosomal RNA. Another one is to enrich for polyadenylated transcripts, which include all of the protein coding genes, the mRNAs for all the protein coding genes, as well as long non-coding RNAs, but it excludes many, many families of non-coding RNAs. There's ways of dealing with PCR bias, as I mentioned. So basically, you could amplify a single RNA molecule over and over again, but having that unique identifier, unique molecular identifier with N and N, can basically allow you to distinguish those. And then when you start adding up all of the single cell RNA-seq data, what you basically see is that it actually starts looking like RNA data, except that you can basically see this dramatic variability from cell to cell. In some cells, for some genes, you see all of the cells having signal. For other genes, you see only a subset. For others, there's zero signal for some and very strong signal for others, and so forth. Okay? And then we can define housekeeping genes, which are the least variable. These are, pretty much on in all of the cells. And then some extremely variable genes between them. And you can validate those using these traditional imaging-based technologies. So fluorescence in situ hybridization can basically tell you, oh, for this gene that is very strongly active across all of these different cells, I expect to actually, when I visualize, see it in all of the cells. For others that are sort of somewhere intermediate, I expect to see this gradient. None of them are really zero, but some of them are higher than others. And then for others, I expect to see zero in some cells and then very strong signal in others. That's exactly what it is. Everybody with me so far? Great. So now, how do we deal with noise in single cell RNA-seq data? You could basically ask about standard metrics of any next generation sequencing technology. You can basically say, what is my fraction of reads that align to the genome? How much of the transcriptome have I covered? How many of my reads are from ribosome versus non-ribosome provenance? How many duplicates are there? How many times did I have a single profile with a single barcode corresponding to, in fact, two different cell types being blended? So this is the duplication rate, or the duplet rate. And also a bunch of very specific single cell metrics. What is the overall complexity of my library? What is the probability of detecting a particular transcript? And how does that relate with the properties of the gene? And you can basically compare that with different technologies, Fluidigm, SNARE-seq, and so on and so forth. You can basically look at what is the fraction of genome alignment, what is the fraction of RNA from ribosomes, where do the genes map? Are they primarily mapping in exons or in introns or in UTRs or in intergenic regions? And what is the overall coverage of each transcript based on the overall expression of that transcript? And you can see sort of this response curve for different technologies and just to compare them. You could also ask, where along the transcript do I cover, do my reads map? And basically, the closer to the 3' end, the higher the mapping. And the reason for that is that many of those are 3' primed. You basically reverse transcribe from the 3' end of the gene in the 5' to 3' direction on the opposite strand. And that's why your coverage is highest here, and then it drops off as you go towards the front. Everybody with me so far? And you can also ask, what is the overall complexity of my library? Namely, as I sequence more and more and more deeply, how many genes do I detect? And you can see here that, for some technologies, I saturate to about 5,000 genes very early on. For other technologies, I saturate more like 11,000 genes. Makes more sense. Everybody with me? And then for the duplication rate, this is sort of this template switching technology that is at the base of SNARE-seq. And then the other thing to recognize is that the biological noise actually depends on the expression profile. Namely, for some cells, you basically have very low probability of detecting the transcript. You have to go to much higher levels, and for others, you detect them much more rapidly. So basically, you'd like to be like that. You wouldn't want to be like that. Basically, you'd like to be able to detect them even for lowly expressed genes. Okay. Now, there's two types of noise in single-cell data. There's sampling noise, namely, I'm sampling cells. I'm not sequencing every single cell. I'm just sampling those cells. And there's also PCR biases, which is more of a technical source. So you could try to decouple the two, basically by asking, what are the ways to eliminate these, these two components. And one way is to focus on very highly expressed genes and then ask, why am I not detecting them in every cell? There are other ways of dealing with some of these noise and some of these batch effects and some of these doublets. One way is actually multiplexing. So this allows you to reduce batch effects, doublets, and also cost. The idea is the following. As you sort of make those bubbles, those droplets, sometimes two cells will fall in the same droplet. And if you only have one sample, every cell has the same barcode. So your doublets will increase as you capture more and more and more cells, because the probability of having two cells in the same droplet is simply increasing as you're saturating the number of droplets that you have. So you could go with one sample for each of those reactions and then increase the number of droplets. And that's basically the reason why many of these experiments stop at about 5,000 cells, because after that, the fraction of droplets increases dramatically, and therefore you have unusable data. However, if instead of putting only one sample in your reaction, you put 3 samples, that means that every single one of those cells could form a doublet with either a cell with the same barcode or with a different barcode, or with a third barcode. So I only have one chance in 3 in forming a doublet with a cell that has the same barcode because it came from the same sample. Everybody with me so far? So that means that the number of droplets that have 2 cells that I cannot tell apart will be reduced dramatically by a factor of 3. If I put 9 different samples with 9 different barcodes each, and then I sort of create those droplets, then I have 1 chance in 9 of randomly mating and sort of, you know, coupling with another one of those droplets. Yeah. So you have individual cells from each of your samples. And now the question is do I profile only this sample and then only that sample and then only that sample? Or do I add my barcodes and then profile combinations of those samples? So this Frankenstein product is not going to be an issue because I have a unique barcode for each of the samples. The only thing is that as I'm adding them all into my suspension, they're basically coming from different, you know, basically I'm mixing in cells with different colors. So then when I have a doublet, if I see red and green barcodes together, I'm like, oh, that's a doublet. I shouldn't study it. Does that make sense? So that basically allows you to dramatically reduce the number of undetected doublets. You still have 1 chance in 9 of pairing up with a cell of the same color, but by sort of multiplexing, you reduce that. So you basically take all of your, you know, samples, for example, 9 different individuals, and then you sort of throw in the cells, and you now sort of throw in the barcodes, and you now have, you know, set of barcodes for each of those. And there's many technologies for being able to label those this way. You know, multi-seq, also very cool acronym, multiplexing using lipid tag indices, allows you to basically barcode things using these unique, you know, lipid tags. And using this, you can basically see, you can basically do an experiment by having 9 individuals in each of your experiments. So you could have multiplexing across different sample types. You could basically say, if I'm profiling schizophrenia samples, bipolar samples, and control samples, instead of doing 9 schizophrenia, 9 bipolar, 9 control, I could actually do 3 of each in each one of these multiplex experiments, enabling me to actually eliminate or at least dramatically reduce those batch effects. And then you can basically ask, if I label these individual cells by sample or by which library I used or by which individual was the donor or by the particular diagnosis of bipolar, schizophrenia, or control, you know, what is that intermixing? And you can basically sort of recognize every single dot here is a cell. And then this is just a 2-dimensional plot that tries to sort of maximally separate the cells that are distinct from each other transcriptionally. And then this coloring is based on the inferred cell type. This coloring here is based on which 10x library I used. This coloring by which individual that cell came from. And this is based on which diagnosis it came from. And here you could say, oh, great, there's some portions of the space that are clearly only one diagnosis, but they're also only one individual. It's very hard to say that this is because that individual had bipolar disorder or because that individual just simply had something weird in their, you know, in the particular sample. Is everybody with me so far? All right, so that's all at the RNA level. There's obviously great interest in figuring out what is the epigenome variation looking like at single cell level. And basically that's what single cell ATAC-Seq does. So this is the assay for transposase accessible chromatin. You basically throw in transposase, you basically ask where does it cut, and every place where it cuts, you basically know that transposase could actually cut in that location, and therefore that it's accessible. You can see here what DNA accessibility looks like using a DNA assay. This is basically cutting the DNA and then asking where is it accessible to DNAs. This is where is it accessible to transposes. And then this is now doing single cell ATAC-Seq. So this is the signal from each individual cell. And you can add up that signal to basically get at the profile of the aggregate of all those cells, which in fact does match the profile of the bulk sample. Okay? Here's one technology, DNA accessibility. Here's another technology, transposes accessibility. And this is the cumulative signal of single cell ATAC-Seq. And then the fun part, of course, is not looking at this signal, because that's the same thing you could get at bulk, but it's looking at the differences between individual cells. Okay? So at every row, you basically see that there's some cells that have more signal here and other cells that have less signal here. Okay? This is very challenging because with DNA accessibility, with RNA, you basically have one shot at capturing RNA with every RNA molecule. And if there's, I don't know, 1,000 RNA molecules of that gene in that cell, you have 1,000 shots at goal. Everybody with me here? So if you're looking at DNA accessibility, how many shots on goal do you have? How many molecules are there? You're looking at DNA. You only have 2 molecules, moms and dads. So you either have 0 reads, which means, oh, that region is not accessible, great. Or you have 1 read, which means, oh, great, that region is accessible only in dad's copy. Or you have 2 reads, oh, great, that region is accessible in both dad's and mom's copy. Okay? But of course, distinguishing 0, 1, and 2 from the sampling stochasticity is extremely hard. So that data is extremely noisy. So looking at any one cell in any one location doesn't give you much information at all. Okay? Simply not seeing something at that one location, that one cell, is not informative. However, you could do all kinds of cool things. You could basically say, let me cluster together 50 cells that are nearest neighbors, and then create virtual amalgamations centered at every cell with their 50 neighbors. You now have 2 times 50 shots on goal. Everybody with me on this? There's another approach that you could use at the single cell level. Instead of adding up reads in the same location across cells, you could add up reads in the same type of region across genomic locations. If you, for example, want to say, is the cell more active for the, I don't know, NF-kappa B transcription factors? Okay? You could basically say, let's find all of the motifs for that factor, and then ask in aggregate across 1,000 different locations, how many reads do I have in that one cell? Raise your hands if you're with me on this one. Awesome. So again, this gives you thousands of shots on goal. Okay? So that basically means that I can study things either by pooling cells or by pooling genomic locations. But at the single location level, it's just simply not very informative. But what's really cool is that I could cluster together all of the cells from the same cell type, and all of the cells from the same cell type, and all of the cells from the same cell type, and then look at an aggregate profile of each cell type in isolation. So if I have a way to aggregate information across each individual cell, for example, by saying, well, in microglia, I expect these transcription factors to be open. And if I look across the whole genome and I find all of the, I don't know, SP1 transcription factor like motif locations, and then aggregate all of their signal, then I can just say quite reliably that this is a microglial cell, and that's a neuronal cell, and that's an oligodendrocyte cell. And then after I do my single cell ATAC-Seq experiment, I can then aggregate the signal for each of the cell types, effectively equivalent to sorting each of these individual sort of cell types, only it's really at the single cell level. And what's really cool is that after I've aggregated them, I can then say not only what is the average signal, but also what is the variation in that signal across cells of the same type. So it gives you strictly more information than sort of first sorting cells and then doing a single experiment which gives you one average value for all of the cells, as opposed to actually being able to look at the variation between cells. Are you with me? Awesome. So how does that work? You basically sort of, you do the sort of microfluidic capture, you lyse the cells, you add these transposes, and then you sort of release it, and then you extend it and you PCR amplify, and you have the individual barcode, and then you sort of label things based on the barcode. the same thing as ATAC, only at the single cell level. Okay? So you can basically now play this game. You can say, what are the transacting factors that are more active in different cells? And you can basically sort all of the cells based on the amount of inferred activity by aggregating all of the locations of GATA1 and JUND and CJUN and P300 and so forth. Everybody with me? So you could also start asking those cells that have high, SMC3, do they also have high CTCF? And you can basically say, how are the coactivity patterns of different transcription factors playing out for individual cells, right? Through their targets. And you can sort of combine the single cell ATAC with single cell RNA by sort of asking how does the transcription of that factor vary across cells, and how does the inferred target activity of that same transcription factor vary across cells. And you can play all kinds of games to understand how is, for example, the hematopoietic lineage differentiating between, myeloid and lymphoid and sort of all kinds of other sources of these cells. And you can ask, how are the transcription factors changing their expression as I go down, and how are their targets changing their activity as I go down these differential lineage. Raise your hands if you're with me so far. Awesome. So you can also start using the single cell ATAC and the single cell RNA together to basically start asking across hundreds or thousands of cells, what is the vector of activity of RNA and the vector of activity of chromatin accessibility looking like, enabling you to now start asking about correlated activity between DNA accessibility and RNA expression, okay? DNA accessibility is down here, RNA expression is down there. For every target gene, you can basically say, what are all of the non-coding regions that turn on in the same cells that the genes are turning on? And how do you do that? Well, you've only profiled RNA in some cells, and you've only profiled DNA accessibility in other cells. So what you need is a way to align the cells together, okay? So one way to do that is to basically perhaps do an iterative approach where you first infer the linking, and then you use the linking to basically say, can I predict the level of RNA expression using the level of DNA accessibility, enabling you to have both RNA observed and RNA inferred from DNA accessibility for every one of those cells. Once you have the same metric space, when you've projected both RNA datasets and DNA accessibility datasets into the same dimensionality space, you can then co-cluster those cells. You can basically co-cluster RNA and DNA profiled cells in the same space. Some were profiled only with RNA, some were profiled only with DNA accessibility. And then look at the nearest neighbors across those. And instead of doing this correlation analysis at the single cell level, you can do it 50 cells at a time. You can basically say, across the entire genome, what is the correlation between, any pair of elements between every pair of cells, or across all cells, what is the correlation between RNA and DNA? Who's with me so far? Awesome. Of course, profiling DNA and RNA separately in different cells has a lot of shortcomings. So there are technologies that are being developed now to profile more than one type of information from the same cell. And there are, some are basically doing DNA methylation and epigenomics, others are doing DNA accessibility and RNA, others are doing, high C and methylation, and so on and so forth, okay? So there's many, many such technologies. These three are the three so far that are doing DNA accessibility and RNA from the same cell, okay? So I'm only gonna highlight one of them, so PAIRED-seq. So basically the idea here is that you're doing your single cell isolation, and then you lyse and physically separate these RNAs, and then you add these unique barcodes, and then from the same barcode, you're profiling RNA and you're profiling DNA accessibility. And in the end, you have matched barcodes that allow you to basically correlate RNA activity and DNA activity in the same cell without having to virtually align yourself together. Everybody with me? Awesome. So, whew, that was just the technology part, okay? It's a fast-moving field. 5 years ago, this lecture didn't even exist. And basically, we went from traditional single-cell analysis to single-cell RNA-seq, different ways of dealing with noise, to a multiplexing approach that reduces batch effects, reduces the number of doublets, but also reduces the overall cost because you're now pulling multiple samples in your each of one of your experiments. We saw single-cell epigenomic analysis and its limitations and the need to sort of pool either multiple cells or multiple genomic locations. And we also saw multi-omics at the single-cell level. Okay, who feels that they've learned stuff today? Yay, great, awesome. All right, let's take a 30-second break. You guys could use some stretching, and it's mandatory. Everybody has to stand up, stretch, get some blood flowing back into your brain. All righty. So now that we have all this data, what are we going to do with all the data? That's, I guess, what you guys are here for. So how do we extract biological insights from single-cell data? So we're going to look at different technological developments on the computational side now for clustering singular cells, singular genes, dimensional reduction, and so on. Okay? So what you could do is look at cell-to-cell correlation, look at gene-to-gene correlation. You can impute missing values, again, addressing this major unique characteristic of single-cell RNA sequencing, which is that you're sampling cells and you're sampling genes within the cells. So you end up with a lot of zeros. Why are there a lot of zeros? One reason for a lot of zeros is that there are some genes that are simply not expressed in that cell. Another reason is that transcription can be bursty. If I know that I'm going to need sooner or later, 50 copies of that protein, I could every hour produce a different RNA. Or it could be like, listen, I'm going to need 50 proteins, by Monday afternoon. So I'm going to make a bunch of RNAs now, and then I'll be ready, and I'll just translate them when I get to it. Okay, it's kind of like doing your homework in batch. So cells do the same thing. They basically transcribe a bunch of one gene, and then a bunch of another gene, and then a bunch of another gene. Okay, so when you sample individual cells, you might end up with a cell that's in the midst of transcribing. You might end up in a cell with a cell that has just made its 50 copies, or with a cell that has already made the protein, it's done, and by now the RNA is degraded. Okay, so part of the reason for the burstiness, for the zero prevalence, and for the sort of non-uniform sampling, is that transcription itself is bursty. So that's a biological reason. And another reason is just simply a technical reason. So you just might get unlucky as you're sampling RNA molecules. Just might not catch RNA molecules from particular cells. So a lot of the techniques that you're going to see have to deal with zeros at every step of the way. And as you start developing your own techniques for RNA analysis at the single cell level, you should basically realize that you have to deal with zeros. Okay. So this is basically from a recent review paper looking at best practices for single-cell RNA-seq analysis. And basically what you have to consider are all of the different steps that you have to go through to analyze the data. So basically, there's the raw data processing, and then you end up with some count matrices of basically how many RNA molecules did I observe for each gene in each cell. Everybody with me so far? So then a lot of things have to happen, okay? Everything we've talked about now has been here, but the computational toolkit is much, much more diverse, okay? So one step is quality control, basically simply saying, how many cells have above background quality. Another one is normalization, basically looking at sort of the number, the count depth or the size, and then sort of normalizing based on that. You can also select features, basically select the highly variable genes and sort of ignore the less variable genes as less informative. Because why would you do clustering using all the housekeeping genes? They're always on. All they're going to do is add noise. Whereas instead, if you focus on only the most variable genes, you might have a better chance of clustering. You can also do a lot of data corrections, so basically correcting for batch effects. So as I showed you earlier, when you get your single-cell RNA-seq data and you label every single cell by the individual it came from, you basically see that there's some green patches and some purple patches that are completely distinct from each other. Whereas if you correct for batches, you basically end up with these well-covered regions that have all of the colors in each of the areas, basically saying that, you know, whenever you sample now, you will have comparable data sets across different individuals. Everybody with me so far? Another big step is, of course, visualization. So laying out all of these different count matrices in some visualizable space. And most of the time, these are 2D. In some cases, you're going to have 3D visualization that basically lay out the cells in some space, and you can rotate that, basically see how the cells relate with each other. 4D and higher are, you know, a little harder. So humans are just very good at 2D, somewhat good at 3D, terrible above that. And then you basically then carry out the downstream analysis, okay? And there's a huge array of things that you can do from that. One of them is basically taking all of your cells and then breaking them up into little chunks. What cell types or what cell states, to get back to your question, does every single part of this two-dimensional space represent, okay? So you could basically cluster them and then define unique colors or look at a continuum between them. So after you've clustered them, you basically can label them by identifying markers that are already known. For example, we know that neuronal cells have mu n plus, and then, you know, I don't know, the dendrocytes have GFP plus, and so on and so forth. So you could basically use that to label which ones of those clusters correspond to which cell types that we already know about. Raise your hand if you're with me. Awesome. So then you could basically say, well, what is the trajectory with which all of these different cells relate to each other? If you're looking at a developmental time course or a disease time course, you basically ask, can I build a pseudo time? Of course, this is not time because you're only doing one snapshot. It's pseudo time because you can basically say, well, if the cells are in different stages of the differentiation, maybe what I've captured is early, middle, late, you know, stage in disease progression or in development and so forth. You can basically infer those trajectories and then understand the dynamics of every gene along that pseudo-time axis and sort of ask about states that are perhaps not as stable. So basically, you know, in here, you might have a stable state yellow and a stable state purple, but in between, there's a lot of, you know, variability. Okay, so you can basically use that data to define that. You could also look at Alzheimer's and non-Alzheimer's individuals and then say, what are the genes that are differentially expressed between them? And there's, you know, additional methodologies for that. And you could also look at the mixing of different cells across different conditions. You can basically say, okay, great, this is my case control for Alzheimer's. But now let's look at tau pathology and how does tau pathology of that individual from which the cell came from relate with the amount of expression or the pattern of expression of that. So you could basically paint your conditions onto that same 2D aspect, axis, basically saying that tau is here and amyloid is here. Everybody with me? Okay, awesome. So then you could basically carry out analysis at the cell level or carry out analysis at gene level, okay? At the gene level, you can basically say what genes are differentially expressed. For example, focusing only on this subset of purple cells, what are the genes that are differentially expressed between Alzheimer's and non-Alzheimer's individuals? Everybody with me here? And then you can basically do this as if this was a bulk sample by averaging the expression of the cell, of the genes in each cell, or by actually using that variability and then using the variance as a way to compare the two conditions, because simply looking at the averages doesn't give you the same statistical significance than knowing that each of the distributions is quite tight between them. Everybody with me? You could also ask at the level of gene sets. So what is the overall change in expression? So basically this is at the level of individual genes, and this might be very noisy. But as we saw with single cell attack, the ability to integrate across many locations in the genome increases your power. So what pathway analysis or gene ontology analysis or any kind of gene set analysis does is you can basically now aggregate information and sort of combine information from many genes of the same type, and then look at how all of these genes sort of together paint a more accurate picture than what you would get from individual genes. And you could also look at overall regulatory networks or protein interaction networks or co-activity networks or co-expression networks, and basically look at modules of closely connected genes in one of those networks and see how are they behaving without constraining yourself to only previously known gene ontology. Everybody with me here? So that's sort of on the gene side. Then at the cell side, you basically do your cluster analysis, you can do your compositional analysis, you can do your cluster annotation, you can do your trajectory, your metastable states, and looking at these expression dynamics across cells. So basically here your dimensions are genes, and here your dimensions are cells. Okay, everybody with me? So now let's dive into individual components of this whole pipeline, basically see how they work. So basically, at the level of genes, cells, dimensionality reduction, and so on. Okay. So first of all, clustering similar cells. So there's a lot of methods and a lot of applications of single cell analysis. One of them is looking at environmental stimuli, basically what happens when I trigger a particular process in the cells, what happens during development, what happens during the cell cycle. So some cells that have just finished dividing or others that are in the process of dividing, how do they change their expression patterns? And also understanding the spatial context of the data. So basically, if you're looking at a brain sample, what's happening in a particular region of the brain is encoded in that sort of actual physical layout of the cells, and you kind of need technologies for doing that. So one technology for looking at the spatial context is SLIDE-seq. So what's SLIDE-seq doing? It's basically using a two-dimensional slide, like from hockey, and you basically lay out onto that puck a set of unique barcodes that are spatially coherent. You now take your brain sample and you basically put it onto that puck. So the barcodes that are already arrayed out are now going into your brain sample and labeling different cells that are geographically, in the same location with the same barcode. Raise your hands if you're with me on this one. Awesome. You can then use that as your first barcode. After that, you can dissociate the cells and then do single cell profiling, where you now add a second barcode for each cell. You basically now have two barcodes, one for the location, one for the single cell. And then you do your overall sequencing reaction, and you can basically map every gene, every transcript to the corresponding cell, and every cell to the location that it was in some two-dimensional space. Everybody with me here? Raise your hand if you're with me. Awesome. Okay. All right, so you can basically do all of these types of analysis, and then you could look for continuous phenotypes of cells, basically what are, pro-inflammatory versus, sort of regulatory cells, or you could ask what are the different discrete cell types, and some of those will be well labeled based on existing markers, but you might also realize that there are some tiny little clusters that were not previously known, and that happens all the time. Every single time you do a single cell RNA-seq experiment, you go deeper than anyone has ever gone with non-single cell RNA-seq technology. So in the brain, even though it's been studied for like almost 100 years, to recognize what are all of the different cell types based on their morphology, based on their activity, based on in situ hybridization, and so on and so forth. There are some very rare cells that no one has actually ever observed before, but you can recognize those using single-cell RNA sequencing, okay? And as you start going to disease states, this is actually a great place to find new cell types. Perhaps they're cell states, but new clusters that are quite distinct from the other clusters that only appear in disease. Again, the folks who are studying disease are different from the folks that are sort of doing these sort of fine-grained cataloging of different cell types. But now you can couple the two with just one single cell RNA experiment. Okay. And again, we talked about this temporal progression across the pseudotime interval, this continuous cycling across different cell states through cell division, spatial positioning, and so on and so forth. Okay. So how do we go from these complex tissues to individual cells? Can we identify what are the cell types or the cell states in a complex tissue? And you could do that in the mouse retina. You could basically look at what are all of the cell types of the mouse retina. In fact, just a few days ago, we published a paper on the first analysis of the human retina, and we were able to identify some cool new insights on sort of age-related macular degeneration based on the cell states and how they differ between individuals, and allow us to map genetic variants associated with AMD to particular regions and cell types of the human retina. But the retina, the mammalian retina, is a fantastic example of the complexity of cell types in the human body. You basically have some cell types that are, across different layers. You basically have extremely rare cell types. You have, these bipolar cells, these photoreceptors, horizontal cells are extremely well laid out. And then you can now start mapping them individually using single-cell RNA-seq. So the first analysis by Makosco et al. Was basically looking at 50,000 different retina cells and then grouping them into different clusters and then identifying what are these clusters, what are the expression levels of those, and how do they map to these classically known lineages of relationships between different cells in the retina. Basically, you could distinguish neurons versus non-neurons based on expression of some genes and so on and so forth. And basically, these maps were drawn over decades. And now you can go back and say, well, here's the map that I get simply by clustering my data. You basically have many more clusters than were previously known. You have the full, hierarchical organization of these clusters, separating them into neuronal cells versus non-neuronal cells, cones, rods, etc. And then for each of those, you also have the cell type markers of what are the signatures for each of those. So, it's a little depressing. Some people spend decades of their lives mapping these individual cells, and now with one experiment, you can sort of, not only recapitulate that, but in some cases go further. And you can basically find 39 new, clusters with many new markers that can now be validated. So again, previously, people had basically selected particular genes for profiling, but now you can go in an unbiased way and say across all of the genes in the human genome, what are the markers that we should actually be using to define those cell types that perhaps we knew about before, and also new cell types that are, newly identified. So that's at the level of cells, basically clustering similar cells. You can also cluster similar genes. You can basically say, what are the most variable genes? And then what are, and for example, housekeeping genes are, much less variable. They show high average, but very low cell-to-cell variability. And then these are at the bottom here. And then these guys have very high variability, and then in general, lower expression per cell. So, and then the variation is where things are interesting. You could also ask, how are cells on or off, genes on or off across different cells? You can basically take the vector of activity for every gene across cells. You basically say this gene is 0 in all these cells, high in all these cells, intermediate in all these cells, and so on and so forth. And you could cluster genes based on their presence absence patterns in different cells. And without even looking at high or low expression, just based on the on-off differences of a particular gene across different cells, you can actually cluster genes of similar functions. You can basically exploit the fact that genes co-vary across cells to start inferring the expression of missing data. You can basically say, well, there's all these zeros, but some of these zeros are because of the burstiness. Some of these zeros are because of technological artifacts, and some of these zeros are just based on sampling noise. I just didn't get lucky when I was sampling to catch that gene. So the question is, are these zeros biologically meaningful or not? And you can do that by, you can address that by basically saying, great, if I look at 10,000 cells, in bulk across two different replicates, I basically have this very, very strong correlation. If I look at individual cells, I have, a big chunk in the middle, but there's many zeros in one cell that are in cell S1 that are non-zero in S2, and many zeros in S2 that are non-zero in S1. Okay? Or you could basically take every individual single cell and ask, how does that correlate with the 10,000 cells? And what you see is that that correlation is in fact much higher, which suggests that it's not just noise in the single cell, it's actually biological variability. You could basically ask how much of my deviation from correlation is due to sampling bias and how much is due to cellular variation. And basically some of these zeros are actually due to undersampling. A very important way and a very powerful way for dealing with these zeros is to basically say, well, if I have a matrix of numbers that is truly coming from some biological process, then that matrix will actually have a true dimensionality, an eigendimensionality if you wish, that is lower dimensional than the, than the 1000 by 1000 matrix of genes. So you could basically say, can I embed this very, very large matrix of gene expression by cells into a lower dimensional space? And what's really cool with doing that is that in that lower dimensional space, you might actually find that you can kind of tolerate a lot of these missing values. How? By basically realizing that there's only so many different cell states, and there's only so many different gene pathways. And therefore, I can take my matrix of gene by cell, reduce its dimensionality, and basically find a set of eigengenes that sort of capture the full diversity of different pathways, and a set of eigencells that sort of capture the full diversity of cellular states. Okay? And basically using that, you can express the original matrix by a lower dimensional projection. And then you could actually multiply across those three matrices of sort of eigenvalues, eigengenes, eigencells, and their mixing components to basically recover per cell, per gene expression matrix, which now has all the zeros filled in, because I was able to infer them based on these, remultiplying out these components. Raise your hands if you're with me on this one. Awesome. Any questions? Great. So now let's see how that applies. So basically, you could, apply this process to basically say, what is this, gene by cell matrix looking like with different levels of, dimensionality reduction. And what you can see here is that, for example, using this particular approach, you could, recover a very clear set of signatures when the original data is in fact, very, very noisy, for example. So here's what, this principal component analysis looks like in a 300 cell data set. You can basically see that, you could label each of these individual cells, and then you can ask, what is the distribution of the first principal component, the second principal component, third principal component across different times of this experiment. Basically, you can look at the response to some stimulation and then say, how are these PCs, working across this time interval? And you can basically see that different PCs capture differences that are very biologically meaningful coming from these cells. Others are basically sort of overlaying all of these different time points, but some are very clearly distinguishable. So the input gene list can dramatically alter the output of your PCA. So basically choose that input gene list wisely. Perhaps you can ignore, like exclude from that PCA analysis genes that are, always zero, or genes that are always on. You can assign, different biological or functional interpretations to these PCs. These sometimes correlate with technical quality. Basically, some PCs are simply reflecting batch, and then not all PCs are in fact significant. So basically, and then there's major limitations that principal components represent a linear combination of these individual features, but some of these combinations might actually be nonlinear. Okay. So you can now start asking, how are these principal components correlating with biology? And what you can see is that, for example, if you look at, these two principal components, you can actually see that they kind of correspond to this movement of cells across differentiation pseudotime, or, across, different aspects of facial recognition. You can also use more complex and more sophisticated models for doing this beyond just simply linear models. You can basically look at negative binomial models that are zero inflated, that allow you to sort of capture this abundance of zeros. You can basically sort of separate out these matrix of genes by cells into both known covariates and how they decompose it, or unknown covariates. And then these known covariates can be either at the sample level. Basically, I know that these samples came from sick individuals or older individuals or males versus females, or gene level covariates that basically say, well, I know that these genes cluster into these gene ontology terms, so I could use those as the basis of decomposition. And I could also discover sort of de novo covariates that are not already in my list. Raise your hands if you're with me on this one. Awesome. And then you could also go a step further and sort of use deep learning as a way to capture these factors. So basically, instead of trying to model them in a purely Bayesian fashion, you could basically say, let's use variational inference at the single cell level to basically learn the distributions that are very difficult to model using these traditional sort of Bayesian approaches, but simply throwing an autoencoder or throwing a deep neural network as a way to approximate these internal distributions. So you could basically sort of look at what results from all that in that fashion. You could also distinguish different cell types by any of these approaches, and then build consensus clusterings from those, and also consensus cell states by looking at the diffusion of your cells across that dimensionality space. Most of the cells are moving this way, but then you see some cells that are sitting in between those states, and you can start interpreting those. So you could also, there's new approaches that are emerging on multi-resolution analysis. So basically dealing with the fact that there's a lot of different cellular states, and then these can be encoded either at the cellular type or within the cell types at the cellular state level, and then building networks that sort of capture the similarity patterns of all of the cells, and then asking in this sort of multi-resolution way, what is the nearest part of the network, what is the diffusion of the network surrounding me, and so on and so forth. So, for example, ActionNet is a new method developed by Shahin Mohammadi in my group that basically tries to sort of capture this across these different resolutions. And you can interpret this variability using known pathways, using trajectories through cellular space, and sort of identifying these trajectories. So again, this is one of those slides that you could spend hours looking at. So, there's an analysis of what are all of the different types of methodologies and how are they doing their learning. And basically, there's a lot of information here that I really encourage you to look at. And then this is sort of a meta view of what are all of the steps that you could take on your single cell analysis. You basically start with single cell expression. You have some way of reducing the dimensionality through similarity, through manifold learning, clustering, or graph approaches. Some are using combinations of both of those. And then you can see kind of like a subway map going through different stations. Each method basically uses some combination of all of these different steps. Again, something that is really worth wrapping your head around and saying, well, if I'm developing methodology, how am I going through this phase? And sort of expressing your own methods in the context of these primitives is very, very helpful in understanding sort of where is the novelty and what is missing and what is perhaps not captured at all by any of these methodologies. Everybody with me on this one? Awesome. So again, we talked about missing data imputation. There's various methods for doing that. A very powerful one is MAGIC by Dana Pereira's group. It's basically looking at this iterative way of learning manifolds onto which you project your data that sort of better capture the true variability. And you can see here that you go from extremely sparse data sets to much more meaningful data sets. And there's a lot of methods for integrating multiple single cell data sets. For example, canonical correlation analysis can work across these multiple data sets, mutual nearest neighbors, similar to what we talked about in gene mapping. And you can basically look at what are the most interesting and most high performing methods across each of these steps of normalization, imputation, clustering, trajectory inference, data integration, and then evaluating the results. And you can see in boxes from a recent review paper, sort of which are the ones that appear to be performing the best. Okay. All right. In the remaining -1 minute of the class, I'm going to take an extra 4 minutes until they kick us out. Tell you a little bit about what's going on right now. And basically, there's a lot of analysis on mouse brain, basically looking at all of the different mouse cell types. A lot of analysis on human organoids, basically being able to grow individual, mini brains from induced pluripotent stem cells of different individuals, and then asking how are the cell types of these organoids matching the cell types in the actual postmortem brain. Looking at the early cortical development to basically see how these organoids are recapitulating this development. And also our own group has done a lot of studies in the variation in those brain samples across different individuals in the context of disease. Again, trying to map what are these intermediate changes that are happening. So this is a paper that we just published this May in Nature in collaboration with Li Huizai, Hans Reddy Matis, and Jose Davila, who were the co-first authors. And basically here, the goal is to look at how are gene expression patterns changing in individual cells for individual genes. And starting with 48 individuals of Alzheimer's and non-Alzheimer's. So these are control individuals who have normal cognition and none of the pathological variables, to early stage to late stage Alzheimer's disease. You can basically ask, if I do single cell profiling of all of those 48 individuals, capture 80,000 cells, about 2,000 cells per individual, what does this tell me about the overall space? And the first lesson is that the overall dimensionality is driven by the cell type. So regardless of which individual these cells came from, you can recognize quite reliably the different cell types. And then within each of those cell types, you can then subcluster the cells into subsets of excitatory, subsets of inhibitory neurons, subsets of astrocytes, oligodendrocytes, microglia, OPCs, oligodendrocyte progenitor cells, and then ask, how are these subsets of cells that are now learned completely de novo within each of the cell types correlating with pathology? And what you can see is that for every one of those major cell types, the subclusters are in fact capturing healthy or sick cells, which is quite remarkable. They also correlate with the BRAC stages of disease progression or the cognitive decline or the overall Alzheimer's variable of pathology or not. What's really fascinating is that they also correlate with sex. Whether an individual is male or female has a huge correlation with whether their cells are coming from the sick cluster or the healthy cluster. Female individuals have many more sick cells,
which is quite remarkable. In fact, the prevalence of Alzheimer's is much higher in women, even correcting for the fact that women live longer. So this might form a basis for why this might be, that in fact, women have much, many more cells from these quote unquote sick states. But what's remarkable here is that we had actually selected those individuals to be matched for their cognitive and pathological variables. So that might actually suggest that women are in fact much more resilient, that even though they have these pathological, these cellular level dysregulations that are associated with Alzheimer's, they appear to be handling it quite well without showing the cognitive and the pathological consequences. You could also start mapping what are the cells that change, the genes that change in expression early in the disease versus late in the disease for each of the cell types. You can basically look at this extremely cell type specific change that happens early on versus later on where you see much more global processes usually associated with stress response, which are very common across the different cell types. You can also find differences between male and female individuals. You see that male individuals, female individuals have about 3,000 genes that are differentially expressed before Alzheimer's, but after Alzheimer's, there's about 6,000 genes that are differentially expressed, suggesting that these male-female differences are perhaps sometimes exacerbated in the context of disease. And in the case of Alzheimer's, of schizophrenia, you can also start looking at these differences across different cells and comparing schizophrenia control and bipolar, and then asking what are the corresponding pathways that are differentially active between all of these cells. The next thing you can do is actually start looking, using the single cell data sequence rather than just expression level, start looking for mutations that are happening in those cells as they're dividing. The concept is that you're starting with a single cell and then you're developing that into a whole organism and a whole brain. And mutations that arise early on will be kept in all of the cells that descend from that lineage. Everybody with me here? So you can look at the sequences of your single cell RNA-seq data to basically find these mutations and then map them onto specific expression patterns. And then sort of recognize mutations that are accumulating in those dividing cells but are not found in these neuronal cells. You can also find that these mutations are much more prevalent in glial cells that are still dividing versus neurons that are post-mitotic, suggesting an accumulation of mutations with divisions. And you also find that if you cluster the cells and you label these disease-associated neurons, they actually tend to have more mutations than the healthy excitatory and inhibitory neurons. And part of the reason here might be simply stochasticity. If you have mutations that randomly hit, that randomly happen more or less frequently, those cells that accumulate more mutations are also the ones that are more likely to become sicker. And that sort of changes the phenotype inference from the individual level and from the brain level to the cell level, to basically say that some cells are sicker than other cells, and those have more mutations. And those mutations cluster in specific genes and specific pathways across different cell types. So for example, oligodendrocytes show more mutations in particular genes, and excitatory neurons show more mutations in particular genes. Again, what will make a gene sicker or not is not somehow driven by selective hit on the mutations. These mutations happen randomly, but if they hit the wrong pathways, then that cell will become sicker. And you can see here that all of these cells, for example, share exactly the same mutation across many cells from the same individual, consistent with this clonal expansion of the mutations. The last thing you can do is basically deconvolve bulk data into single cell data to basically infer QTL effects at the single cell level across thousands of individuals. So basically for every individual, you can basically ask what is the impact of each genetic variant at each cell type for each cell, for each gene. And you can basically infer genes that change more, so genetic variants that change expression in inhibitory neurons or in microglia or in astrocytes, and then correlate that with particular disorders, and then start mapping the disorders that are genetically driven by expression differences in different cell types. So what we talked about is number one, single cell profiling technologies from the traditional to the very modern. Number two, different computational techniques for extracting biological insights from the single cell data. And number three, a case study focusing on the human brain and looking at both the cell type diversity and functional diversity, initial maps of brain diversity, and then brain variation at the single cell level in Alzheimer's, schizophrenia, and bipolar, as well as somatic mosaicism and deconvolution. So a lot of these studies are in fact work that is still unpublished. Much of that is coming from our own lab and many collaborations that we have. So this is an extremely active field, a growing field. I really encourage you to embrace it because that's really the future of where biology is headed. Who feels that they've learned stuff today? Yay, awesome. All right, guys, see you on Thursday, and this will not be on the quiz.